In this series of videos, we're going to be learning about what mutations are, at least in terms of the genetic definition, and clarifying some of the misunderstandings or outright misinformation that exists regarding mutations in the public consciousness. Now, the rather unimpressive definition of what a mutation is genetically is any permanent change that occurs to an organism's DNA sequence. You'll note that we've referred to DNA sequences rather than genomes because even though mutations that specifically cause differences in protein structure and function are the most interesting ones to talk about, that's not to say that mutations only occur in genomic DNA. It's possible for a mutation to occur in any DNA sequence. So what exactly causes mutations? The reality is that the overwhelming majority of mutations actually occur spontaneously because a mutation, given that a mutation is simply a change to the base pair sequence in DNA, a lot of the time mutations simply occur because DNA polymerase 1 and DNA polymerase 3 make errors when they are undergoing DNA replication, meaning that when DNA is duplicated, sometimes a DNA polymerase molecule will put in the wrong base when copying DNA, resulting potentially in a permanent change that occurs to DNA. Now, the causes of mutations that more people in the collective consciousness are familiar with are mutations that are caused by chemicals or energy that we call mutagens. So, as mentioned previously, some of these are chemical in nature. For example, a very toxic and mutagenic substance is a chemical called benzene, which you'll recognize from the benzene ring here, which has the ability to insert itself between DNA base pairs and disrupt how DNA replication occurs, but a lot of the time they are energy-based. So probably the most well-known examples of forms of energy that can alter DNA are forms of radiation, such as ultraviolet light that is broadcast to Earth from the sun, gamma rays that are a very harmful type of radiation common with nuclear isotope decay, and X-rays, another high-energy form of radiation that's used to take X-ray pictures whenever you have a bone analysis being done. Now, we're not going to talk too much about the mechanism of how mutagens work because each individual mutagen has specific effects depending on how it interacts with DNA. But what we can do is look at the step-by-step process of how a mutation is actually created using a spontaneous mutation caused by an error of DNA polymerase 3 when it inserts the wrong nitrogenous base into a DNA sequence. And if we look here, this is our template strand or the parent strand here, and we can see that at this location, which is a G, the DNA polymerase 3 molecule has inserted the wrong base accidentally, thymine, when a cytosine should have been here instead, and on the corresponding daughter strand, we can see that on the template strand C, a G was put in, which matches the template on the other side. Now, if you were to call this as a mutation, you would actually be incorrect because this change is not permanent yet because your cells have specific enzymes that can actually look over a replicated DNA strand and proofread the DNA looking for any DNA damage such as a wrong base pair that's been inserted. So we've talked about DNA polymerase 3 and DNA polymerase 1. DNA polymerase 2 is actually a specific DNA polymerase molecule that has proofreading abilities, and one of those abilities is to detect when a wrong base has been inserted simply because the hydrogen bonding between bases doesn't line up the way that it's supposed to be. The problem is that the proofreading of DNA occurs after DNA replication has already finished, meaning that the daughter strand already extends all the way to the end of the parent strand. Now, we as humans have brains and analytical ability, so we can identify relatively easily that this is the parent strand and this is the daughter strand. But if you are a DNA polymerase molecule, all that you can see is that there is a replication error that has occurred here because of irregularity in hydrogen bonding. There is no way for you to identify what the parent strand is and what the daughter strand is, and therefore 50% of the time this mutation will be fixed correctly. So the way that DNA proofreading enzymes work is that they actually identify a mismatched pair and then pick one side of the DNA strand, essentially randomly, and cut out an entire section of DNA and then re-replicate that strand, hoping to fix the error properly. Now, 50% of the time, this problem is fixed properly because the DNA polymerase molecule will choose the proper side as the template strand and re-replicate the daughter strand with the correct base in its place. However, 50% of the time, the proofreading enzyme will actually choose the wrong side of the DNA double helix and actually cut out the original parent strand here and treat the error as the new template, and therefore insert a base that corresponds to the wrong base here. And this, when we have done this, results in a permanent change to the DNA sequence, because now, is there a way for a DNA polymerase proofreading molecule to recognize that this is an error? Well, again, the answer is no, because all that it can tell is A is supposed to pair with T, and there is no error in that. It doesn't know what the original template strand looks like, because again, it's just an enzyme. It has no collective memory of what the original sequence was. And when we have made a permanent change by firstly introducing an error in replication and then fixing, quote unquote, the wrong side of that replication error, this is when we can truly say that we have a mutation to the DNA sequence, because there is no way for your proofreading enzymes to even recognize that this is a replication error, and therefore no way in order to reverse this. And once again, the takeaway of this is that mutations occur because enzymes, your proofreading enzymes, do not know which side of a DNA replication error is the actual source of the error, because they cannot tell the difference between the parent strand and the daughter strand. In the next video, we're going to start looking at the consequences of what even changing a single base within a DNA sequence can do, and what potential consequences it might have if that DNA sequence ultimately codes for a protein.